afternoon, all of you. Till now, I discussed about various compartment models, right? So, to analyze the pharmacokinetic data, you have three different kinds of compartmental kinetics. So, first one is compartmental model. In that, we have seen about mammillary model and quaternary model, right? These two methods we have discussed in the last class. Then next model is physiological model and non-compartmental analysis and non-compartmental analysis. See, ma'am, for determining the pharmacokinetic parameters, okay, for determining the pharmacokinetic parameters, you need to calculate various kinetics and dynamics from these kinds of models. So what we have learned in this compartment model, your body should be assumed as a compartment, okay, it will be consisting of more number of organs or tissues. So you can assume it as a single organ or tissue as a peripheral compartment. So there is a movement of the drug from central to peripheral compartment. And one more important point, your compartment may be arranged either of parallel or of serial manner. So that much we learned in these two, mammillary and quaternary. And then coming to this physiological model, so how it will be explains you to calculate the pharmacokinetic data. See here. So physiological models, they are also called as PK models. Physiologically based pharmacokinetic models. This is physiologically based pharmacokinetic models. And then they are more realistic as it is based on known anatomic and physiological data. The name itself is also indicating this data will be based on anatomic and physiological data of individual organs. See, organs with similar perfusion. That means, suppose if your liver, kidney, these are having some similar perfusion, same perfusion rate. So you just group these kind of organs in one group. You just make them into one group. See here, organs with similar perfusion is grouped in a single compartment. This is a single compartment. Then like lungs, liver, brain, kidney, these are all grouped as rapidly equilibrating tissues. Why? Because these are of highly perfused tissues, your lung, brain, kidney. So you just assume these all highly perfused tissues are grouped at one group. Okay, and then while muscles, adipose tissues, these are slowly equilibrating tissues. Equilibration is nothing but when the organs or tissues will shows high perfusion, they are having rapidly equilibrate. That is of they are having rapidly equilibrate with either of ADME processes. So in such cases, these muscles and adipose tissues will be slowly equilibrating tissues. That is called SET. So the organs or tissues such as bones that have no drug penetration. See, you have liver, brain, kidney. These are of highly perfused tissues. These are also rapidly equilibrating tissues. And muscles and adipose tissues, you have slowly equilibrating tissues are there. And in case of bones, they doesn't have any drug penetration property. Okay. See here how the orientation of this physiological models, how it will be organized. See ma here, this is a lung and this is a heart. See, this is a gut, this is a liver, and this is a kidney, and this is a liver, lung, and brain. So these are of highly perfused tissues. So these may be joined to the venous blood, and here you have arterial blood. So the movement of the drugs will be taken place depending upon the size of the tissue or volume of the tissue. So how it will be dependent upon? Suppose if your heart is having some small area to drug absorption, so and if your liver is having some high area to absorption your drugs, if suppose I am telling you, so in case of heart there is low penetration of your drug is there. In case of your lungs, here it will be having high drug penetration. So it will be totally dependent upon the volume of the tissue fluids and the size of the tissue. So the movement of the drug in these kinds of physiological models will be dependent upon the size of the tissue and volume of the tissue. So these kinds of tissues will have highly perfused tissues. So these may be attachment of arterial blood flow and venous blood flow. So in this is an input, and this is an output. So this is nothing but the elimination, and this is nothing but the absorption. So here you have absorption and here you have elimination. So the drug movement between these organs, or tissues is noted by your physiological models. And then you have sixth or seventh unit, I could not remember exactly, you have separately physiological models are there. In that you need to learn some derivations of the equations. In this unit 2, this matter is enough, and in that you have to learn various advantages of these physiological models. What is the major advantage? Here you not require any mathematical expression. You should not require any mathematical expression to calculate the PK parameters, right? Then how you need to calculate? See here, data fitting is also not required. Data fitting is also not required. And then, so the PK models can be easily calculated depending upon the size and volume of the organ. Size and volume of the organ, which will be clearly obeys the plasma drug concentration curve. Plasma drug concentration curve. What we learned previously, you already know the plasma drug concentration curve. And then the most important point, by the use of these physiological models, you should attain the correlation of data. You should attain the correlation of data several times. So for whom you should correlate this? With the humans and with the animals also. So these are of some advantages of physiological models. See ma'am, there is no mathematical expression to explain your PK kinetics, PK pharmacokinetic parameters, right? And there is no data fitting is also not required. And the third one is the PK parameters can be easily depending upon the size or volume. You can calculate the size and volume of the organ, and it will be clearly obeys your plasma drug concentration curve also. And the most important point, you should attain correlation between the humans and animals. If you can calculate the data for 4 times, 6 times, 10 times, it will give the same results. There is no manipulation and there is no less results or more results. It will give the accurate results. Okay, accurate results between the humans and animals. Which results? These all are pharmacokinetic parameters results. So this is of some advantages of physiological model. Then the only disadvantage is it will be whatever the whatever the data is obtained, that will be expensive. Sorry, exhaustive. Exhaustive. This is only one disadvantage of this physiological model. That's why the people are not majorly considering this physiological data, and we could not easily give any justification also. Why? Because in case of compartment models, you can assume it your drug is moving from 1 to 2, so that you can give some mathematical expression, then you can calculate very easily, right? So then in case of physiological derivation, highly perfused tissues will be joined to that less perfused tissues, and how we can achieve that based on volume and size of the organ. It is somewhat difficult to attain some pharmacokinetic parameters also. That's why it is rarely used, but the compartment modeling is commonly used to calculate PK parameters. These physiological models are of two categories: perfusion rate limited and diffusion rate limited. See here, perfusion rate limited. These are also called as blood flow rate limited. It is based on the assumption that the drug movement within a body region is more rapid. That is, its rate of delivery to the particular region by the perfusing blood. So, if your organ is having enough perfusion, then it should be assumed as having rapid rate of delivery of the drug into the plasma. Then, however, it is only applicable to the highly membrane permeable drugs. So, what are the types of drugs? Low molecular weight, poorly ionized, and highly lipophilic drugs. So, the drugs will have thiopental, lidocaine, etc. These will said to be perfusion rate limited. Because of their lipophilicity, those are not having enough solubility. So, definitely there is a limitation of that perfusion rate in these kinds of drugs. So you just remember the two drugs examples, thiopental and lidocaine. In case of diffusion rate limited, it is more complex and is applicable to drugs in which the cell membrane acts as a barrier. See ma here. Here you have diffusion rate limited or membrane permeation rate limited models are there. In that, this is more complex and is also your cell membrane acts as a barrier. So which kind of drugs will be transported with the use of this diffusion rate limited? Highly polar, ionized and charged drugs. I think you already know the diffusion phenomena in case of absorption. So the phenomenon is the same as that of absorption. And then distributed parameter model. This model is not commonly used, and this is not in your syllabus also. This model is analogous to the physiological model. This model is specifically useful for assessing regional differences in drug concentrations. So you don't focus on more this. And then what are the various applications of these pharmacokinetic models? It is helps to understand the behavior of drugs in the patients, and it will be helpful for predicting the concentration of drugs in various body fluids. Okay, it helps optimizing the dosage regimen, and it is also useful to calculate the toxicity, and it should useful to correlate the plasma drug concentration with the pharmacological response, and it is useful for determining the influence of altered physiology on ADME, and it explains the drug interactions also. See how many applications are there, or how many advantages are there by knowing these kinetic models. So it is very simple. Once you go through it, you just remember what the points I highlighted here: behavior of drugs, predicting the concentration, optimizing the dosage regimen, and analyzing the toxicity, and correlating the plasma drug concentration versus pharmacological response or therapeutic response. And it is useful to calculate the physiology on drug ADME, and it is useful to detect the drug interactions also. So these are of number of applications which will be by your pharmacokinetic models. And this is the last one. This is the third model to analyze the PK data. This is the last topic in your second unit, right? So the non-compartmental analysis is also there. The name itself is indicating these are not following any compartments and all. That's why this is also called as model independent. That means it is not following any model. See, it follows linear kinetics, so it can be applied to any compartment model. So it will always follows like this linear kinetics. So it will be applicable to any type of compartment model. It is based on the statistical movements theory. See ma, here this is very very important. And one more important thing, this statistical movement theory is also present in unit number 7. So in that unit I will clearly explain to you statistical movement theory, which involves the collection of the experimental data following a single dose of the drug. First, I will show you the graph, then I will tell you what is it. See, ma'am, when you take the graph between the time versus concentration here, this is the Y and this is the X, no? So, after administering a single dose of the drug, first it will give curve like this. Then again it will give curve like this. In this statistical moment theory, I am telling you. Okay. So this is the area, you know, like AUC, right? So this is the area of the drug which will be present in our plasma. That is nothing but area under the curve. And then what do you mean by this? You will get one more curve also, no? So here. So what do you mean by this? This is called as AUMC. AUMC is called as area under the first moment curve. Area under the first moment curve. Okay. So this is the area under the curve. And in this statistical movement theory, it will follow trapezoidal rule. See here. Here some blocks are there under AUC. See, horizontal and vertical lines are there. Under AUMC, you have only vertical lines like this. So this is called AUC and this is called area under the first moment curve. So it will follow trapezoidal rule. See here. This will give statistical movement theory will give the MRT. MRT is the mean residence time of the drug. That means how much time your drug will be resides in your body. It explains you the residing of your drug in your body. So MRT is the mean residence time. These are very very important. MRT is mean residence time. That is equals AUMC divided by AUC. AUMC is the area under the first moment curve, area under the curve, or also called as area under zero moment curve. So this AUMC is obtained by plot of product of plasma drug concentration versus time. See, AUC is obtained by plotting concentration versus time. Is it right or not? What I told you, the sentences are same like that. So here, there is no need of learning all these derivations here. I will tell you in the seventh unit. You just know the statistical movement theory and you just know the MRT equation. MRT equation, mean residence time is equals AUMC divided by AUC. Area under the first moment curve. This is area under the first moment curve. And AUC is area under the zero moment or area under curve. So this is what this MRT will says you. And then these are all simple derivations, ma. See here, in this trapezoidal route, this graph will explains you. See ma, here this is the time. This is the zero time and this is the zero concentration, right? So you just observe this graph. This peak, where you will attain this peak, that peak will be the standard line. You just consider that peak will be the standard line. Then this first peak line, dotted line will indicate you at 0. That is nothing but T0. Why T0? Here in the x-axis, we can take time as a point right here. So that's why this is called small t. And why I have written 0 here? The peak where it will attains the graph, then that is called as initial time. See here, 0 time. Then after that, where that peak will be declining. See here, the variations in the peak. This to this peak, the variation in the peak is some different, no? See ma here, suppose the graph is like this, up to that, then it will be like this. So where the variations will you find some difference in the graph? So you just put it the marks where you find the difference like this. So then you just join the dots to that x-axis like this. So first graph you should put it 0, then next one is 1, next one is 2, next one is 3. These are the common numbers I have written here. And instead of writing direct numbers, you can write T0, T1, T2, T3 like that. Why? Because this is of time on x-axis. That's why you have write like this T1, T2, T3, T4. Then with respect to that T1, T2, T3, T4, you can write the concentrations of C1, C2, C3 and C4. Then if you want to calculate this AUC, you just find out the difference between T2 minus T1, C1 plus T2. See here, you are subtracting the time from T2 minus T1, and here you are adding the concentration. So this much of concentration will get this much of time. This much of time it will be utilized to movement of this much of concentration. You just write it down. Once you practice writing, then it is very easy. Then same like that, here AUC T4 to T3 also you have to calculate it. Then this is the area under the first movement curve. So this is the first movement curve. You need to calculate the concentration and time from 0 to infinity. Okay, this is the first moment curve. You need to calculate from 0 to infinity time. That's why this is 0 to infinity and C dt into dt, Ct into dt. So this is what the final AUC equation, and this is what the final AUMC equation. See ma here, in case of AUMC, small t came here. In case of AUC, there is no t here. That means, what does it mean? AUC, area under the curve, this curve will travels only according to the concentration, not with respect to the time. In case of first movement curve, it will be reflected onto the time. That's why the time will be here, Ct into dt, and here C into dt. So that is the major difference between AUC and AUMC, and this will be explained you only by the use of this statistical movement theory. Okay, so this, from this you completed the second unit of introduction of pharmacokinetics. So what I discussed you till now in this pharmacokinetics topics, first you need to focus on the definitions. You need to focus on the definitions. Once again I am telling you, this chapter is very important one, and very easy to learn also. Then the relationship between plasma drug concentration versus time curve. Everyone has to learn this. I will ask after the first mid exam. Okay. Then you have to learn compartment models. Compartment models, mammillary and quaternary. And then you have to learn physiological models. And then non-compartmental analysis. Okay, in this physiological model, again it will be present in your 6th unit, and non-compartmental analysis, it will be present in 7th unit. So in this second unit of this BPPK, you just concentrate on definitions, plasma drug concentration versus time, and compartment models. These three are most important. Okay, so from this, I completed your second unit of this BPPK.